right, guys, let's start. Can you hear me in the back? Oh, jeez. Let me try to. Okay, good. Okay, so we continue now. I mean, the topic that we. So there is I'm only alone here, and there are so many of you, so it would be a little hard to overscreen. All right, so we continue the topic that we started last lecture, the enzymes. So, and perhaps, you remember that the enzymes, it's one of the one of the landmarks of all sort of living organisms, because these are the biological catalysts which make reactions which otherwise would take forever or will not happen at all, take place in a biologically meaningful time frame so that the living organism can exist. So essentially, the living organism exists largely due to the existence of the enzymes. So last time we were talking about how the enzymes work, and today we're actually going to spend a little bit of time on studying how, the enzymes kinetics. This is probably the only mathematical part of this course which will be in the exam, of course. Okay, so what the enzyme kinetics? So kinetics is a study of the rate at which the compounds react. So you probably already had this partially that, kinetics in one way or another during your inorganic chemistry courses. So, rate or velocity is the amount of product accumulated or substrate consumed per unit of time. So basically it's how many moles of product appeared, over some certain amount of time. So rates of the enzymatic reactions, are affected by, of course, the enzyme concentration itself. So the more the enzyme, the faster the reaction will go. However, as we talked previously, enzymes being catalysts, being true catalysts, right? So they're not used in the reaction. So technically, one molecule of an enzyme can crunch infinite amount of substrate. So the rates, of course, depend on the concentration of the substrate and factors such as temperature, pH, ionic strength, and different regulators, which we'll talk in detail today. Okay, so the simplest way to write down the chemical reaction catalyzed by the enzymes would be if we'll take the as simple as possible type of reaction when there is one substrate. So when there is not several substrates, but just one substrate that the enzyme acts upon. So the enzyme plus substrate reversibly turns into the enzyme substrate complex, which then decomposes to enzyme and product. So again, in this scheme, we have only one reactant, one product, and no inhibitor. So these are just some assumptions. So substrate is consumed during the course of the reaction, obviously, right? Because the enzyme acts upon it so that if there is no constant supply of the substrate, so the amount of the substrate decreases while the amount of protein increases. So last time we had this very simple formula that the velocity or the rate of the reaction proportional to the rate constant times the substrate concentration. Because K is constant, substrate decreases, right, over the course of the reaction, so the velocity also decreases as the reaction goes. Then if we want to study velocities, we need to sort of make some sort of agreements, right, so as to when and what kind of velocities we're going to measure. So if the velocity itself is it's a value that changes over the course of the reaction, we need to have some agreements in terms of at which time point we need to do it. Let's just imagine a very simple experiment. So we just mix an enzyme plus substrate and record the rate of the substrate disappearance or product appearance as function of time. So that would be the speed of the reaction or velocity. So the initial velocity is just, the tangent to the curve taken at time 0. Okay, so on the x-axis it's time, right? On the y-axis it's the amount of the product that appeared. Okay, let's focus on the lowest concentration of the substrate, right? So obviously the reaction starts at some speed, but then the speed goes down. So again, the tangent or the slope of that curve at each given time point is the velocity. So it's basically the amount of product divided by time. So as you can see, as the reaction progresses, I mean, the slope constantly falls, right? So initially it has the highest velocity, but as the reaction goes, I mean, the velocity goes almost to like a plateau, which means the slope is zero, the velocity is zero. So every reaction slows down over time because the amount of substrate decreases and the amount of product increases. Okay, so if we will increase the amount of substrate, right, progressively, so we'll see that the speeds, of course, will be high because the more the substrate, I mean, the higher the velocity. The main question is here, okay, what if we will increase the substrate concentration infinitely? Would the velocity increase also infinitely? The answer is no. So the more the substrate we will give, so the less and less the velocity of the reaction, the speed of the reaction, or the rate of the reaction, of the enzymatic reaction, will depend on the concentration of the substrate. In other words, there is some certain amount of substrate, above which adding more substrate does not, make any difference in terms of the rate. So the rate stays the same. So basically now imagine, that the like infinite concentration of substrate, which is somewhere here at the ceiling, right? So, but then it will still go at some certain slope, which will be only marginally more than this tangent. So let's now put it actually in a more tangible form. Okay, so this velocity or the rate of the reaction at the infinite concentration of substrate is called Vmax. So it's the rate above which increasing the concentration of substrate has no effect. So, so the plateau here, oh, it doesn't work, okay. So the plateau here indicates that it indicates that the enzyme can be saturated with the substrate. So we'll see this actual plateau on the following slide. And formation of the enzyme substrate complex must be the part of the catalysis. So we'll focus on this part in a second. Okay. This is how it looks in terms of the graph. So again, now if we will plot the initial velocity, again, this is only the tangents, or the slopes of the curves in the time point 0 against the substrate concentration, we will see that as the substrate concentration increases, right, the velocity, basically, or the rate, first it grows, but then it starts to grow slowly, and then at some point it becomes like steady. So you will increase more, you will add more substrate, but the speed will not change. And it will be asymptotically trying to reach this Vmax limit. So this is the maximum speed which the enzyme is capable of reaching. So okay, so the lots of assumptions, in this field, in the field of the enzyme kinetics, and lots of breakthroughs were made by these guys, Leonard Michaelis and Mott Menton. So, and quite often, this enzyme kinetics is referred to as Michaelis-Menton kinetics. So, and we'll see what their assumptions are. So first we consider that the one substrate and one product reaction. It's the simplest. So we have one substrate, one product. Again, this is the right way of writing this equation, so where we can see the reversible formation of the enzyme substrate complex and reversible decomposition of that complex into the enzyme and product. So product is like the concentration of the product is negligibly small in the very beginning of the reaction, right, when the reaction just started. So that we can actually almost like remove this reversible component of that reaction, because again, so in the very beginning of the reaction, because the very little product, right? Because of that, the association of the enzyme and product reversibly to the enzyme substrate complex is almost nothing. So that's why we can sort of ignore that component. And then the decomposition of the enzyme substrate complex into enzyme and product, if it occurs more rapidly then it's vice versa. So if the enzyme substrate is formed more rapidly than the enzyme plus product, so then we can consider that this step would be a rate limiting step. And as a result, a rate limiting step is the one which determines the rate of the whole process. So that what leads us to write that the initial velocity simply depends on the rate constant 2 and the concentration of the enzyme substrate complex. So in the textbook, I mean, there is a nice way, I mean, how that equation from this form, which is non-usable in practice, it's like, because we cannot measure we can measure velocity, right? Because we can measure the amount of product formed and we can measure time. But there is no way we can measure the ES, actually. So this is a very transient state, as we know, so there is no way of measuring that one. So this equation needs to be turned into like more practically usable form. And that's what actually the Michaelis and Menton did. So they turned that one into the so-called Michaelis-Menton equation. So this is actually how the final equation looks like. It has two forms. You can write V0 like K2 times enzyme total concentration times concentration of substrate divided by the sum of Km, that's a constant, plus the concentration of the substrate. Or these like two members can be joined together, and actually that's what it's called the Vmax. So either this form or that form, so you can use either one of them. Okay. So this equation describes the ideal rate of enzymatic reaction. So it basically expresses V0, or like the initial velocity, in terms of the parameters and can be measured experimentally. So what do we have here? We have concentration of substrate. So that's very, very easy to measure, like for any given reaction. So, so the important assumption that we need to make is that it assumes the initial steady state. So is that we're talking about the very, very beginning of the reaction when the enzyme substrate complex and the substrate concentration has not changed significantly. So that's just the very beginning of the enzymatic reaction. So here we have actually, perhaps, I mean, the most interesting thing here would be this Km. So this is a Michaelis constant. So this is a concentration of the substrate at which velocity is 50% of the maximum. So it's kind of an approximate measure of the substrate affinity for the enzyme, but we will actually, talk about this later. So mathematically, Km is just the ratio of the rate of the ES breakdown, so to ES formation. That's what it is. It's just the ratio of the rate constants. Again, Michaelis-Menton equation describes the ideal rate of the enzymatic reaction. And it's applicable only for one substrate reaction. So again, we're talking about the most simplistic case. So, and it describes relationship between the speed of the reaction, the maximum speed of the reaction, and the initial substrate concentration. So you may actually ask whether this equation fits any experimental data. So let's go back to this graph that I already showed you, right? So the dependence of the speed or the rate of the reaction from the substrate concentration. Okay, first let's look at this infinite substrate concentration. At the infinite substrate concentration, the V is actually almost constant, so it tries to reach the Vmax. So, and indeed, if we will try to go back to this equation, right? So let's say the S is much more. Let's say, so the S is infinity, right? So then it would be much more than Km. And in this formula, you can just simply cross them out. So you will see V equals to Vmax. So that seems to be right. Okay. Another thing. So what will happen with that equation if we will look at the concentration of the substrate close to zero, like when the substrate just been added to the reaction? Again, so let's take a look. Okay, when the substrate concentration is much, much smaller than Km, right? So then V equals to Vmax divided by Km times S. So it turns into a linear equation. So it's a linear plot. And indeed, so in the very beginning of that plot, I mean, there is a zone which looks almost as linear. So, and that is what we refer to as this initial velocity. The most interesting is when the concentration equals to this, arbitrary parameter called Km, or Michaelis constant. So it's when S equals Km, then the velocity equals to half of the maximum velocity. That's the definition of the Michaelis constant. Again, Michaelis constant is a concentration of the substrate at which the reaction rate reaches half of the maximum. So you will have this kind of problems in the homework or in the exam. And so if the question clearly asks, can you calculate the Km for that given reaction? So if you will give the answer like whatever number followed by nothing or followed by the units, which makes no sense, let's say second minus first degree. So that is totally incorrect. So the units of measure of Km is the concentration units, because by definition, Km is a concentration of substrate at which Vmax, at which the rate of the reaction is half of the maximum. So make sure to include the correct units. So because that actually, I think, I mean, it's more important that the units would be right rather than the number itself would be right. Because if the units are wrong, means that you do not comprehend, like you didn't get it. So if you have a problem, I mean, just make sure to come to my office hours or to TA's office hours, where we'll be, we can go over again this topic again and again. So how to determine this kinetic parameters such as Km and Vmax? So as I said, the power of the Michaelis-Menten equation lies in the fact that it allows to experimentally measure everything. So we can measure the speed and we can control the concentration of substrate, because I mean this is the experimentally kind of controllable parameters. So how to determine this Vmax and Km, because it seems like these are the most important parameters of the given enzyme. Of course, I mean what we can do is to actually get a series, or let me just show you the one which looked simpler, like this one. We can get a series of these graphs, right? So how would you work from here? Again, you cannot do the infinite concentration of substrate, right? Because mathematically we can talk about infinity, right? But practically no. So then we would not know exactly where the Vmax value is located, right? Because we will try to kind of draw the line, but what if that line is a little bit higher, a little bit lower? So depending on where this line is, right, so you will actually calculate half of it and get the Vmax. So, and again, because I mean it's very steep in the beginning, so the small arrow in determining of the Vmax level will be translated into a huge arrow in the determination of the Km, because you will actually, so let's say, I mean, this line would be a little bit higher, right, and the half of it would be here, and then the Km would be almost double of what is shown here. So what I'm trying to say, if we will be operating only with this direct plot, so we will not be able to get the these values of Vmax and Km accurately. And the solution was mathematical and was relatively simple. So if any of you took the math course, maybe you will remember this interesting trick. So when you actually invert the left and the right side of the equation, it's called double reciprocal coordinate. So when you actually sort of invert to the -1 the left side and the right side. So, and then you decompose them in the members of it, and it will look like a linear, basically, linear plot. So we have 1 divided by V as one member, so then Km divided by Vmax, and then reciprocal concentration of the substrate would be the variable, and Vmax to the -1, that would be the last member of that equation. So if we will plot this, so this trick is called actually Lineweaver-Burke plot. So, and then this type of plot turns into a nice line and intersects with the y-axis and with the x-axis, will tell you very important parameters. So the intersect with the y-axis will tell you 1 divided by Vmax, from which you can easily get what the Vmax is, and very accurately. So, and so will happen with the x-intercept. So here you will get what the negative 1 divided by Km is, from which you can easily deduce what the Km value is. So this will give you much more accurate estimate or determination of these parameters. So as I said, so Km is a concentration of the substrate at which the velocity reaches half of the maximum. So in some cases, it's an approximate measure of substrate affinity for the enzyme. as I said. However, so what the affinity is? It's a dissociation constant or association constant, right? So the Km would be this type of association constant only if this member, the K2 rate constant, will be absent. So it will be absent only when it's much smaller than the K minus 1, right? So in other words, when the rate of the decomposition of the enzyme substrate complex is much slower than the rate of formation of the enzyme substrate complex for the enzyme and substrate, right? From here. So in other words, when this reaction go many times before one reaction of that kind will take place. In that case, the K2 would be much smaller than the K minus 1, and the Km turns into just the association constant, which is a measure of the substrate affinity for the enzyme. So basically, so what is the measure of substrate affinity? It's like how much the enzyme likes its substrate, like how much it likes binding it, or in other words, how much energy is being released upon binding of the substrate to the enzyme. Okay. Here just, some examples of what the values are. So here are just some enzymes like hexokinase, the substrate. And of course, we know that the hexokinase has like two substrates at least. It has ATP and glucose. Let's not talk about fructose now. And we can see that basically for each of the substrates, there would be separate Michaelis constant. So it would be 0.4 for ATP, it would be 0.05 for glucose. Okay, they're enzymes and substrate which have relatively high Michaelis constant. Again, high means actually bad in this case, because once again, so the Km is the concentration at which the enzyme reaches half of the maximum speed. So the higher the number, so that means that the higher the concentration needed to be so that the enzyme will reach half of the speed. So the lower this number, the better for the enzyme. So the most efficient enzymes will be the ones with the relatively low value of Km. So because, they can reach half of the maximum at a very low concentration of the substrate. So again, so these are roughly the values, which, the enzymes can have. But what I would like to draw your attention to is here again. So that the unit of measure of the Km is the concentration. So it's molar, millimolar, nanomolar, micromolar, whatever. Another interesting parameter, it's the so-called kcat, so a turnover number. So it's a number of substrate molecules that are converted to product during a given unit of time on a single enzyme molecule when enzyme is saturated with a substrate. So here, basically it's how many molecules of substrate the enzyme can crunch per unit of time, per 1 second, for example. Usually it's measured in per second. So we can see that some enzymes like catalase, they have like insane number of cycles per second, right? While some other ones have relatively small number of cycles per second. So does this mean that one enzyme it's like more efficient than the other? So neither this parameter kcat nor the Michaelis Menten constant can serve as a sort of unique measure of the efficiency of the enzyme enzymatic activity. However, their ratio, which is called the specificity constant, is like a good estimate of that. So in this case, we can see the because the enzymes can use different strategies. Some enzymes have relatively simple substrate, so the Km for the substrate is super high. That means that it doesn't really like its substrate. It doesn't bind with high affinity. But it can crunch many molecules of such substrate per second. So there could be another type of enzymes, like, for example, acetylcholine esterase, which has super high Michaelis constant, right? That means that it's really very specific to its substrate. It really likes it. It binds it with high affinity. But the turnover number is not so great. However, when we actually calculate the ratio of these two values, so we'll get an ultimate indication how good or bad is the enzyme. And we can see that when we calculate that ratio, that is enzyme catalyzed, which has an insane turnover number, it's not as efficient actually as acetylcholinesterase, which crunches much less number of molecules of substrate per second. Okay, so so far we've been talking about a very, pretty, not very rare case, but only about the single substrate enzymatic reactions. And what happens actually if there are two substrates or more in the enzymatically catalyzed reaction? Let's say if the enzyme catalyzes the merging of the two molecules together. And here we actually, can use as an example our favorite hexokinase, so which again attaches the phosphate group from ATP molecule. It transfers it onto the molecule of the glucose on the position 6. So unlike one substrate reactions, for two substrate reactions, different kinetic mechanisms are possible. So we'll see what are those. So what is kinetic mechanism? It's the order of binding of substrates and the release of products. So just going straight. Okay, what is possible? So in other words, we can have enzyme. So it first binds to the substrate 1, or alternatively it can bind to substrate 2, and then, so basically we will get the ternary complex. So it's a complex between the enzyme and both of the substrates. So S1 and S2. And only in that case, we will get to the products. So this is the case with the, let's say, hexokinase. Either this one or that one would be the case with hexokinase, because both of these examples, random or ORAT, so they involve ternary complex formation. So the difference between random ORA and ORAT is that here it doesn't matter whether the S1 binds first or the S2 binds first, so it doesn't matter. So they can bind in any ORA. But in the ORAT example, so then the substrate 1 needs to bind first, then the enzyme substrate 1 complex is formed. That predisposes the enzyme or forms a new binding pocket for the substrate 2, so that then substrate 2 can bind. And this ternary complex can be formed, which is then decomposed to the products. Because this is usually the case with the enzymes where the two groups are chemically merged together or transferred. So there are examples like here, for example, enzyme reaction in which no ternary complex is formed is when a group first transferred onto the molecule of the enzyme, and then from the enzyme it's transferred to the molecule of the substrate. So in this case, no ternary complex is formed. Ternary means free, right? So what are the three? So enzyme, substrate 1, substrate 2. So these are the free entities that we refer to as a ternary complex. So how we can determine what kind of kinetic mechanism we're dealing with for the particular enzyme? So if we do that set of reactions, which that type of experiment that we already talked about, let's say we just simply measure the speed at which the substrate disappears or the product appears, and measure the initial velocities, and then plot it using the double reciprocal coordinates. So we can see that as we will increase the concentration of the substrate 2, if the lines will be intersect, let's say in the zone, then it's just a mathematical solution, a mathematical interpretation of how it looks like. So if this Lineweaver-Burke plots will intersect, then that's basically an indication of the ternary complex being involved in the reaction. While in the case of no ternary complexes involved, the lines will be parallel. So we will have the lines basically going closer and closer to 0 as the concentration of substrate 2 is increasing, while we fix the amount of the substrate 1. Okay, so so far we talked only about how enzymes basically, like the basic enzyme kinetics. And now let's talk about enzyme inhibition. So we know that the enzymes are regulated, so in the living cells, right? So the regulation can occur actually in a controlled way, or sometimes, there are different toxins and poisons, that can basically inactivate the enzymes against the need of the particular organism which has that enzyme. So first, let's talk about the irreversible inhibitors. So what are those? So these are types of inhibitors that chemically react with the enzyme, and they cause irreversible inactivation of the active site of the enzyme. So in one inhibitor molecule can permanently shut off only one enzyme molecule. So, and these are usually powerful toxins and can be sometimes used as drugs. So what would be the examples? Our favorite chymotrypsin, remember from the last time. So, and we remember this serine 195, the one which has this nucleophilic serine group, right? So there is a compound called D-isopropyl. So, okay, this is isopropyl. So there are two of these groups, fluoro, and this is phosphate. D-isopropyl fluorophosphate. So this compound, if added to the chymotrypsin, what will happen? The serine 195 will nucleophilically attack onto the phosphate, right, and will get itself permanently attached to it. So this adduct, it's like basically will not be. I mean, it's irreversible reaction. So this enzyme molecule will be permanently inactivated. So it become inactive anymore. So this is what is like a good example of the irreversible inhibition of the enzymes. So another example. So peptidoglycan. So this is an important component of the bacterial cell wall. So and there is an enzyme called transpeptidase, which links the peptidoglycan precursors into a larger polymer. So here just an example. So we have this enzyme transpeptidase. So what happens is that it actually gets itself. I mean, it also has a serine, which has this nucleophilic OH, which attacks onto the carbonyl carbon group, right, and gets itself attached to that precursor. So this is how the cell wall is synthesized in bacteria. I mean, this is one of the many reactions, of course. I mean, this is not the goal of this particular lecture to talk about how the cell wall is being synthesized. So this is basically like the same reaction in detail. So we have basically one. I mean, we have an enzyme carrying this, and then another peptidoglycan also attacks onto this carbonyl carbon, and we got actually a longer chain of peptidoglycan in the cell wall. The antibiotic molecules, which everybody knows, which belongs to the penicillin class, such as penicillins or ampicillins, and such, they also called beta-lactams. So because of this interesting ring here. So they recognized by the active site of this transpeptidase enzyme. And what the enzyme is doing, it's serine, it's active serine in the active site, tries to attack onto this carbonyl carbon. It does it successfully. And then the resulting adduct, I mean, again, becomes, makes, I mean, this enzyme permanently inactive. So this is how the penicillin and ampicillin type of antibiotics work. So they irreversibly inhibit inhibit enzyme transpeptidase, which is a crucial enzyme for synthesis of the cell wall. Why this makes these antibiotics, work, sort of what makes them to work inside our bodies? The main reason why they work is because bacteria do have cell wall and we don't. Let's say our cells do not have cell wall, so therefore, regardless of, basically these compounds will not have any target in our cells. So, and that's why they target only the bacteria. Okay. Yes. Oh, well, I mean, this is a question for several lectures. I mean, so that's, there's several ways. I mean, just to make long story short, let's say you've been exposed to a poison. What would you do? Like several logical things that you would do. Huh? Huh? No. Let's say you have very limited time to do it. So you will not make it to doctor. Huh? I'm talking, imagine yourself a bacteria, let's put it that way. Imagine yourself a bacteria and you, let's say, floating, right? So you're not like a human being. You're exposed to a poison, right? So what would be the logical things that you can do? Yes? Huh? Die? No. Okay, well, bacteria don't have antibodies, but I mean, this is, I mean, the right track. I mean, keep... Purge, exactly. Pump it out. So that's one way, one mechanism of resistance to many drugs. So it's actually like there are a number, actually, there is a number of these pumps, like the Tol-C, for example, in the membrane of bacteria, which non-specifically pumps out all this trash, I mean, that is around them, so including the antibiotics. And another thing, logically, again, so you can pump it out, so get rid of it, right? Physically, it gets in, but you get rid of it. What else you can do with this poison which is around you? Destroy it. So you can destroy it. You can sort of come up with a gene or another enzyme that will recognize, I mean, let's say this, penicillin, and will chemically destroy it. I mean, the bazillion of mechanisms how that can happen. And probably the third logical thing, I mean, of course, there are more mechanisms, but what would be the third logical thing that you can try to, which is the hardest to get? What does this mean, tolerance? Yeah. What would you change? Let's put it that way. Okay, so first we change the membrane. So we have, let's say, a special pump installed, right? So like a sump pump, which just excludes this drug. Second, we obtain a special, let's say, kind of like, I don't know, life vest or something, I mean, that destroys, I mean, this, the poison itself. And what would be another participant here, right, on which you can act? Actually, the target itself, we haven't talked about that. Let's say, in this case, it's transpeptidase. There are multiple targets in bacteria. So you can alter it. Let's say you can change the pocket where that serine is located, right? So that this drug cannot get inside anymore. So this is also what the bacteria do. But again, it's a very, very long topic, actually, how bacteria evolve resistance. Yes? This is irreversible example. Yes. So that after after this adduct is being formed, right, there is no way back. So essentially, the enzyme, the transpeptidase, is fooled by the chemical nature of this of the penicillin. So the transpeptidase thinks that it's attaching the precursor of the cell wall to itself, right? But in reality, I mean, it's like a Trojan horse. Okay, so let's talk about these were irreversible inhibitors, right? So which are permanently inhibit enzymes. And these are, again, as I said, I mean, so these, this has no connection to the regulation of the enzymatic activity in living cells. It's more related to the toxins, so that some organisms make some, let's say, chemical compounds which irreversibly inactivate the enzymes in the other organism, and that's how they actually fight with each other. However, when we want to talk about the regulation inside a particular organism, right? So we need to talk about reversible inhibition. So which means, which is already in its name, so means that it can be on and off. So what are reversible inhibitors? So they bind and can dissociate from the enzyme. So they're structurally similar to substrates or products and can be used as drugs to slow down particular enzymes. So the same capacity for being used as drugs. So there are several types of reversible inhibitors. They can be competitive inhibitors, uncompetitive inhibitors, and mixed inhibitors. So usually, actually, I like to bring the following analogy about the inhibitors. Usually, also, when we talk about the enzymes, I mean, so what would be a good example of the substrate and the enzyme? So I don't know, I always bring the example of, let's say, chocolate and the mouse. I mean, so you, like, your mouse is like an active site of the enzyme which crunches it, right? So, and the chocolate, it's a substrate. So what would be the example of the competitive inhibition? Even before we talk about, like, mathematical aspects of it and like go into more details. Competitive inhibitor, it's something that looks like a chocolate, but is not a chocolate. Like, my kids have these games, I mean, where they make hamburgers from plastic toys, right? So that would be an example. I mean, they have candies, of course, made from plastic. So it looks like chocolate. It does look like a chocolate, but if you try to put it in the mouth, it doesn't work out. So that would be a competitive inhibitor. So it's something which does look like a normal substrate of the enzyme, which can bind to the active site, but cannot be acted upon due to its properties. I mean, on the other hand, would be uncompetitive inhibitors. It's something which does not, does not necessarily need to look as a substrate. It binds not even in the active site of the enzyme, binds elsewhere, but somehow affects the ability of the enzyme to act upon. Again, in the case with the chocolate and your ability to chew it, it would be a very scary scenario, let's say a tranquilizer drug, right? So your mouth, you have still, I mean, the jaws, the teeth are in place, they potentially can chew it, but due to the tranquilizer, which immobilizes the muscles, I mean, everything in place, active site is intact, but cannot do the job because, I mean, the muscles do not move. In the case of the enzyme, so the active site is actually not perturbed. The substrate can still bind, but the enzymes cannot work on it. Let's talk more specifically about this type of inhibitors. Again, first, I mean, and the most popular is the competitive type of inhibition. So we have the same equation of the enzyme plus substrate turns into the ES complex, enzyme and product. So, and basically we have an inhibitor. Because inhibitor binds to the same place as a substrate, so to the same active site, essentially what happens, it decreases the effective concentration of the enzyme. So it makes, it decreases the number of the enzyme molecules capable of actively crunching the substrate. So, and these basically two complexes would be alternate to each other. So this complex between the enzyme and the substrate is mutually exclusive with the enzyme and inhibitor. So let's just, I mean, main features again. So the competitive inhibitors bind to the active site and compete with the substrate for binding. So they inhibit, obviously, binding of the substrate because they prevent the normal substrate from going in the same place. And they do not affect the catalytic function. So, and what would be the changes in kinetic parameters? So there would be no change in the Vmax because the maximum velocity is a consequence of the catalytic activity. Since the catalytic function is not affected, then the Vmax is not affected. Basically, it only increases the apparent Michaelis constant. So it would look like the Km is higher than it was without the inhibitor. So that's how it would look on the plot, which we'll see in the next slide. Some examples of this type of inhibitors. For example, this actually that molecule is an inhibitor, and this is a normal molecule. It's called dihydrofolic acid. So, so this is the substrate for the enzyme called dihydrofolate reductase. So this enzyme, what it does, it actually it basically is important for transfers of the single carbon elements. I mean, in the bacteria. So these reactions are specific to bacteria, though, and the methotrexate. So it's a competitive inhibitor of DHFR. If we look more closely in it, we'll see it kind of looks very similar, but not exactly like. There are some groups which have been messed up a little bit. And because those groups are not exactly the same, that makes, I mean, this enzyme, it makes it impossible for this enzyme to act upon such a molecule. As a result, it gets into its mouth, but it gets stuck there. So it does not, it prevents enzyme from working. So if we look now at our favorite Lineweaver-Burke plot, or the double reciprocal, coordinates, we will see that actually as we increase the concentration of the inhibitor, let's say the slope, of that line will go up. That means that this thing will become smaller and smaller, closer to zero. And because, again, it's a reciprocal, that means that the Km becomes higher and higher and higher. So that the more inhibitor we add, so they all intersect at the y-axis. That means that the Vmax is not changing. But as we increase the inhibitor concentration, the Km would be rising. So again, the Km as a measure of the affinity of the substrate, which makes logical sense. So the more these plastic chocolates will be there available, right? So the harder would be to actually get the correct one. The real chocolate. To randomly grab it. Okay. Another type of inhibition is the uncompetitive. It's when basically the inhibitor binds. So we have an enzyme, right? So besides the site for the substrate, it has some other site, so which is elsewhere on the enzyme molecule. Doesn't matter where. So this is just a schematic. So the point is that when this inhibitor binds to such enzyme substrate complex, so this enzyme substrate complex loses its ability to catalyze the reaction. So the substrate still binds, but cannot be converted. As you can guess, in this case, both the Km and Vmax would be changing. So the uncompetitive inhibitors would bind only to the enzyme substrate complex. So because it makes no sense to bind to just the enzyme. So because it binds at the spot which is other than the substrate binding site. So they do not bind, that's the key difference with the previous type, do not bind to the active site of the enzyme. And they do not affect the substrate binding itself. So they inhibit the catalytic function. Again, the competitive inhibitors do the opposite. They do affect substrate binding, but they do not change catalytic function. And here is the opposite. As a result, actually both the Vmax and Km change, but the same number of times. And as a result, the lines would look parallel. They will not be intersecting at the same y-axis, so which means that the Vmax is changing. So the Vmax goes down. So the higher the concentration of the inhibitor, the higher would be this negative Vmax. That means that because it's a reciprocal value, right? So the Vmax itself would be lower and lower and lower. And the same will be happening with the Km. Okay, so. And of course, there are mixed type of inhibitors which can basically combine the two features of the two of the enzymes that we talked about. So the mixed inhibitors, they bind to the enzyme with or without the bound substrate. They bind to the regulatory site and inhibit substrate binding and the catalytic function. So the changes in the, in terms of changes in the kinetic parameters, so they would decrease the Vmax, so the maximum velocity will be lost, and they would be changing the Km. However, we don't know exactly how it will change, either making it larger or smaller. So in the double reciprocal coordinates, the mixed inhibitors would look, the effect of the mixed inhibitors would look like this. So the lines would not intersect at the y-axis, but they will intersect somewhere in this zone. So if by doing the experiment and studying the particular inhibitor, so the scientists got this type of representation, this is a key example, this is a clear example of this mixed inhibition. However, most of the, or many of the known inhibitors belong to this class, these competitive inhibitors. So they're easier to find, they're more abundant, and so they're easier to study them. And of course, all of this type of compounds, originally they come from Mother Nature, but they have actually lots of use in medicine. So because we have lots of enzymes, which control the key vital functions of our body, right? And ability to inhibit them when they're not needed is important. So this is why, I mean, the inhibitors of different enzymes have, I mean, lots of applications in the in pharmaceutical industry. So, okay, here's just a summary of what is changing. I mean, during different types of inhibition. So when we have just no inhibition, we have arbitrary Vmax and Km. So if we have competitive inhibition, Vmax stays the same. Michaelis constant increases. That means that the affinity of the enzyme to the substrate decreases. Okay, when we have the uncompetitive inhibition, we have both Vmax and Km change, so they both decrease by the same amount, by the same number of folds. As a result, the line looks parallel. In the case of the mixed inhibition, so Vmax and Km change, but different. I mean, so if Vmax changes, let's say 5-fold, then the Km can change 3-fold. Okay, so the last topic for today is the regulation of enzymes. So one type of the regulation that we already just talked. I mean, this is the inhibition. So, and how else enzymes can be regulated? So they can be regulated by non-covalent modifications or covalent modifications. So here again, there could be irreversible or reversible modifications. Also, the very important type of regulation called allosteric regulation. So it's a non-covalent modification and regulation of the enzyme via binding of a modulator. So here how it can be actually visualized in a schematic view. So we have the enzyme, so catalytic part and regulatory part, right? So M is a modulator. So let's say when there is no modulator, the enzyme is not working. So, but when the modulator binds, the enzyme changes its conformation so that it can bind the substrate and basically exert the catalytic reaction that it's supposed to work on. So this is an example of what the allosteric regulation is. Allos means elsewhere. So that the regulator binds not to the active site, not to where the substrate is supposed to bind, but elsewhere on the enzyme and activates or inhibits it. Again, a subtype of the allosteric regulation would be uncompetitive inhibition, where the inhibitor binds elsewhere and affects the activity of the enzyme. Here's just one of the thousands, if not tens of thousands, of examples of allosteric regulation of enzymes. So this is an enzyme called aspartate transcarbamylase. So it catalyzes the following reaction. But the reaction itself is not so important. What's important is that this reaction is in a long pathway towards synthesis of pyrimidines. So in a couple lectures down the road, we'll see that the pyrimidines are these molecules which serve as precursors for RNA and DNA synthesis, such as CTP, for example. So cytosine triphosphate. So the end of this pathway, if we'll continue it, would be CTP, or cytosine. So the most classical example of the regulation of the enzyme is when the enzyme is regulated by the final product of the pathway. So yes. That's exactly the reason why it's. Oops, sorry, one sec. That's exactly why the wording that is used is modulator, right? So this is kind of doesn't specify whether it's it can be either activator or inhibitor. So, and that's why I said, I mean, a subtype of this allosteric regulation would be uncompetitive inhibition, right? When that would be the case when inhibitor, which binds elsewhere, not on the active site of the enzyme, inhibits it. But the molecules that activate it. So modulator, it's kind of it's a abstract molecule, which, I mean, it's like a collective sort of image of the molecule that can either activate or inhibit it. Again, so in this example, what we talk about the inhibition, usually the enzymes which participate in different biochemical pathways are inhibited by the final product of the pathway in which they participate. So why it's needed? So the cell always needs to control that the synthesis of the particular molecule stays at the, basically stays at the needed level. So that when there too many of them are produced, right? So the enzymes which participate in that should somehow know about that and stop doing what they're doing. So, and this is just a good example. So this enzyme, aspartate transcarboamylase, again, it does not synthesize CTP, but it's in a pathway which leads to synthesis of the CTP molecules. Okay, it has two conformations: the inactive, or so-called T state or closed state, or active, R state. So what happens is that when the enzyme, like in this form, which is active form, produce too much of its product, then its product in due turn led to production of more and more CTP. Then the CTP, when it reaches some threshold concentration, it binds to this enzyme and closes it. So the enzymes become inactive. So later, and the whole pathway stops basically. So the pathway stops producing the CTP. So later, when there is a demand for more CTP molecules, so the ones which actually already bound to that enzyme, they dissociate. So see the CTP molecules, there is a binding sites inside that enzyme. And the binding site for the CTP are not where the active site is. So they just change its conformation from active to inactive. So when there is a demand for more CTP molecules, so they dissociate, just because, there is not enough. Their concentration is too low. They dissociate and turn in this enzyme back into the active state. So being in the active state, it continues synthesis of this product, which leads to more CTP production. So this is a example of the allosteric regulation, and also an example of inhibition of activity of the enzyme by the final product. So in many biochemical pathways, which this is a trend. So whatever is the final product of the pathway usually controls the activity of the enzymes on this pathway. Okay, so the kinetics of allosterically regulated enzymes usually differ from Michaelis-Menten kinetics, and it has this so-called sigmoidal shape. So the enzymes actually basically they go from the highly active state to the low active state. These were examples of reversible actually inhibition or activation of the enzyme. So now let's talk about an interesting case of the irreversible covalent modifications of the enzyme. Let's say our favorite chymotrypsin, before it becomes active in the intestine, is usually secreted as an inactive chymotrypsinogen, right? And the only difference between this inactive form and the active is that just the inactive form has 15 more amino acid residues on the N-terminus, which sort of work as a plug and close the active site of the enzyme. Any ideas why we do not need why we cannot just have the active chymotrypsin right away? It's a peptidase, right? So it's one which chops off proteins. And it's synthesized in the cytoplasm of the cells. If it will be synthesized as an active form, it will immediately start basically digesting the cytoplasm of the cells in the pancreas in whose cells it was produced, right? So you want to make sure, basically when you, in a factory which produces explosives does not want those explosives to explode, right? So there should be some ways of keeping it contained. So this is like basically it's weapons of mass destruction. So that the cells wanted these weapons to be deployed only when they're in the intestine, right? So once it gets inside the intestine, so there is another basically the effector which is trypsin, which cuts off this little part of the chymotrypsin, making it, rendering it active, and so on. So this is a common trend with most of the intestinal peptidases. So they usually synthesized in the cells in this precursor form, so which is inactive, and then only once they get into the intestine lumen, only there they actually turn into the active forms by the partial proteolysis. In other types of reversible covalent modifications of enzymes which affect the activity, perhaps the most common and the most widely used by many different cells would be phosphorylation. So the enzymes, basically there are other types of enzymes called protein kinases, which transfer the phosphor group from ATP molecule onto the enzyme, getting this phosphor form of the enzyme. So this dramatically changes usually the physicochemical properties of the enzyme, because phosphate group is negatively charged, and also it's important to know which amino acid it's attached to. It can be only serine, threonine, tyrosine, and in very rare cases it can be histidine. So, regulation by phosphorylation, this is again one of the most common types of regulation of the enzymes inside the cells. So for many of you actually who will become doctors, some of you who become doctors will be maybe specializing in cancer. And actually this is one of the main reasons and causes of cancer. So because the enzymes which control the cell cycle work through this cascade. And those are the enzymes which usually start to malfunction. So, and actually lead to cancer. Okay, so in other types of modifications of the enzymes that can change the activity would be adenylation. It's attachment of the adenosyl monophosphate groups, or acetylation. So, or even myristylation, we will see this is attachment of the hydrophobic anchors. This is, we will see in a few lectures down the road. I mean, this is the way how some particular, not only enzymes, but proteins can be tethered to the membrane, can be targeted to the membrane. Another type is ubiquitination. So perhaps, I mean, you already heard it maybe in some other biological courses about this type of modification. So ubiquitin is a small protein which is attached by special ubiquitin ligases to a given protein or enzyme. And if there is only one molecule of ubiquitin, it's treated as a modification. But if there are multiple, it's like a death mark. So if this ubiquitin is attached, I mean, then this protein or enzyme or molecule to which it's attached, I mean, is actually being sent to degradation. Okay, so, and from this table, I really want you to remember some very key aspects. I mean, so that that phosphorylation, I mean, so this is the main type of, like the most common way of regulating the activity of the enzymes and various proteins inside the cell. Just an example of how that works. So, so the phosphoryl groups are transferred again from ATP to the regulated targeted enzyme by the protein kinases, which are enzymes themselves. So they just specialized. So to transfer the phosphor group from ATP molecule to the particular serine, threonine, tyrosine, or histidine in the target enzyme. And the attachment of this phosphor group, I mean of the phosphate group to the target enzyme, affects structure and catalytic activity of the regulated enzyme. And as a result, the enzyme becomes, attachment of the phosphate group, again, going back to your question, doesn't mean that it will be inactive or active. It can be either way. So in the, let's look at the example here. So attachment of the phosphate group to the enzyme called glycogen phosphorylase makes it active. But it, again, for this enzyme, it makes it making it active. For some other enzyme, it will make it inactive. So, the fact that it's phosphate is attached doesn't mean one or another. So it would be specific in each case. So we will actually, you will actually talk with the, it will be in the third module with Professor Jeffrey when you start studying glycolysis. So this is one of the reactions that leads to glycolysis. So glycogen phosphorylase basically chops off one glucose units from the glycogen, right, and sends them into the glycolysis cycle. So the enzyme which does that is glycogen phosphorylase, I mean the inactive form, is being activated by phosphorylation through this phosphorylase kinase. Again, so this is an enzyme which attaches phosphate group to this inactive form of phosphorylase. So when these phosphogroups are attached, phosphorylase becomes active and starts basically chopping off glucose from the glycogen. So this is one of the this is a very important reaction in our body because that is controlled by adrenaline. So what happens is that this phosphorylate kinase enzyme is controlled by the cascade which comes from adrenaline. So when the animal sees a predator, it becomes scared. Immediately, adrenaline is being actually sort of secreted into the blood. So when adrenaline reaches liver, for example, right? So the cascade leads to the activation of this phosphorylate kinase, and it starts producing this active phosphorylase and glycogen phosphorylase enzyme, and that prepares the animal for the long run. So it starts chopping off the glucose from the stored glycogen, right? As a result, I mean, it can get lots of energy very fast. So, but we will talk about that when we'll get to the sugars and lipids. So these are very interesting and important reactions. I mean, which is also an example of regulation of enzymes via reversible phosphorylation or dephosphorylation. So the key thing here is that these phosphates can be removed. So and they're removed by the enzyme called phosphorylase phosphatase. So phosphatase takes off the phosphates. So as you can guess, I mean, if the phosphorylase will remain active infinitely, it will very quickly use up all the stored glycogen, and basically the animal will be too much exhausted. I mean, and that is not desired. Okay, so a couple of summary points about the enzyme kinetics that we talked about today. So the enzymatic reaction rates are dependent on substrate until saturation occurs. So in saturation, there is no dependency on the substrate. So we talk about reactions in a so-called steady state, which is when the enzyme substrate and substrate are relatively constant, which is only in the very beginning, right? So when the reaction just started, and we can assume that the decrease in the substrate concentration is almost negligible. So Michaelis-Menten equation or plot allows the key kinetic parameters to be determined. So the Michaelis constant can indicate substrate affinity, while the turnover number, or kcat, is the single molecule reaction rate. So the ratio of kcat to km is a useful measure of an enzyme catalytic efficiency. So the enzymatic reactions that involved more than one substrate, so which are more common, so have ternary or no ternary complex formation. So, and that can be determined through the kinetic mechanism. So by drawing or by actually obtaining those double reciprocal plots, we can determine, the scientists can determine which mechanism they undergo. Okay, enzymes can be regulated. So that's, I guess, one of the important conclusions from today's lecture. So that unlike inorganic catalysts, the enzymes can be turned on and off when needed and when not needed. Okay, so the enzyme inhibitors can be several different types. So they can be competitive, uncompetitive, or mixed. So competitive inhibitors do not affect catalytic function, but bind to the same place as the substrate. Uncompetitive, vice versa. They turn off catalytic or affect catalytic function in a negative way, but bind elsewhere, not in the catalytic center. Again, the mechanisms of inhibition, whether it's competitive, uncompetitive, or mixed, can be determined using these double reciprocal coordinate plots. So the same as the mechanism, determination of the mechanism of reaction. Okay, so phosphorylation of a specific amino acid residues, which we now know are serine, threonine, tyrosine, or sometimes histidine. It's a most common way to regulate enzyme activity. So many proteolytic enzymes are synthesized as inactive precursors. We actually didn't talk about the name zymogens. So it's like chymotrypsinogen. So this is this type. So which are activated by cleavage of small peptide fragments. And that is needed to prevent their premature activity in the cells where it's not needed. Okay, that's it for today. Today, sorry. Homework will be on Blackboard today. Sorry, actually, I'll put it today. I have it. It's already ready. Thank you.